So the second type of substitution mutation is a nonsense mutation. Now, as it says, after the mutation has occurred, the amino acid that used to be coded for is now changed into a stop codon. So remember when we looked at protein synthesis, and you might remember that stop codons are a triplet of bases, like any codon, but what they do is they stop a protein being synthesized. So they stop it being produced. So wherever they are, they will stop that process. And this can then obviously put in the wrong place. This can result in a protein which is going to be shorter than normal, because if you put the stop codon earlier in the sequence, then it's going to stop the process earlier, and so your protein that's getting produced is not going to be as long because it's getting stopped early. So let's just have a look at an example. Here again, we have our DNA sequence, our original DNA sequence. We've got our amino acids that each of those codons codes for. Now we have the mutation has occurred. So you can see the mutation is this cytosine has mutated, and it is now T thymine. So again, substitution mutation, because one's been substituted for another. However, this is a nonsense mutation, and so this codon, TAG, is now codes for a stop codon. And so, because it does that, well, because it's a stop codon, it basically stops the process of protein synthesis, and therefore no more protein synthesis will occur after the stop codon. And so you can see, whereas this would have been a much longer protein, this protein is going to be much shorter because of the stop codon. The third type of substitution mutation is this one here, a splice site mutation. Now, it says splice site mutations result in some introns being retained and or some exons not being included in the mature transcript. So if you remember back to when we looked at RNA splicing, and you'll remember that during splicing, exons are kept in and introns are removed from the sequence. So introns are non-coding regions, exons are coding regions. And so you get rid of the introns, we keep the exons and splice them together for it to be our mature transcript. But if we have a splice site mutation, then this, as it says, this results in some of those introns either being kept in the sequence, or some of the exons, which should have been kept in, are not going to be included. What can this result in? So if a mutated splice site is rendered non-functional in this way, one or more introns may be left in the modified mRNA. Okay, so what happens if we have introns in our mRNA? Well, the mRNA may be translated into an altered protein which does not function properly. So if your mRNA does not have the correct exons, or it has introns as well as exons, both of them are results of splice site mutation, then again, the protein might not function properly. And so here's a couple of examples. We have our exons, introns, exon, etc. A correctly spliced mRNA is you get rid of these introns and you just have the exons spliced together. However, if the mutation occurs, then we can have you can see here one of the exons, exon number 2, is not being included. Or down here, you can see that we have all our exons, but we also have an intron which has been included. So either of these could result in a protein which doesn't function properly.